What is trachoma? It is a chronic infectious keratoconjunctivitis caused by Chlamydia trachomatis, whose diagnosis is above all clinical. It begins during childhood and may be cured spontaneously or recur and lead to the adult complications of palpebral retractations, which in turn leads to entropion of the upper eyelid and trichiasis that causes progressive and irreversible opacification of the cornea. It is a contagious illness, with flies and soiled linens constituting the principal agents of transmission. The diagnosis of trachomatous conjunctivitis is made by everting the upper eyelid and examining the tarsal conjunctiva with the help of a magnifying glass. Clinically, the classification of Macallan has been replaced by the simplified trachoma grading system, with 5 grades to facilitate the epidemiological study of the disease. Follicular trachoma occurs in the young child and is characterized by the existence of more than 5 translucent or yellowish follicles, at least half a millimeter in diameter. Intense inflammatory trachoma is characterized by at least 5 follicles and very red thickening of the conjunctiva that masks more than half the vessels. Follicular and inflammatory trachoma are also called active trachoma. In the scarring stage, or scarring trachoma, which occurs after repeated infections, lines of scar tissue appear on the conjunctiva. In the entropion trichiasis stage, or trachomatous trichiasis, at least 1 eyelash rubs against the eyeball. Finally, corneal opacities cover the entire pupil. The germ responsible for trachoma is Chlamydia trachomatis, a 300 nanometers microorganism with serotypes A, B, BA and C. Its intracellular development makes it difficult to study. The genus Chlamydia has a unique life cycle in the cells they infect. The cell phagocytizes elementary bodies, which differentiate to reticulate bodies. The reticulate bodies then divide in a vacuole, which progressively enlarges to form intracellular inclusions. The reticulate bodies condense to form new elementary bodies, and after 48 hours, the inclusion bursts, destroying the host cell and liberating the elementary bodies, which in turn go on to parasitize new cells. In practice, how do we determine the presence of chlamydia in patients with trachoma? There are different types of tests one can use. The question is, what is the infrastructure and what is the budget available for this kind of diagnosis? The first possibility is to do a gem sustain, but the efficacy of this is weak, not even half the efficacy of culture or immunofluorescence. Second choice being immunofluorescence. For this, you need slide, fixers, alcohol and acetone, extremely difficult to transport to the villages, as the bottled products are inflammable, especially in the heat. There remains the possibility of showing that the bacterium circulates, even if the germ studied is dead. And this is why we chose PCR. It is a genetic amplification. We didn't choose culture either, because with chlamydia, the specimens have to be kept in liquid nitrogen transport media, or at -80 degrees. This is unrealistic. Useful only for very few theoretical studies in conditions of wealth with freezers, liquid nitrogen and hospitals at hand. If we are in the field, in villages where prevalence studies are being conducted, we must limit ourselves to finding the bacterial genome, which we can do using classic PCR or a real-time PCR analysis. We chose the PCR in real time with a Pacman probe so as to always work within closed tubes. This way there is no risk of cross-contamination, no burst of electrophoresis, and no additional procedure to perform after amplification, since the instrument, the robot that does the amplification, detects the amplicons that were amplified and have produced a fluorescent signal within a closed tube. The bacteriologic analysis begins with a sampling performed in the field under strict conditions in order to avoid any contamination. 2 people are needed, a sampler and a porter. The sampler, equipped with sterile gloves, everts the eyelid of the patient, applies the Dacron swab to the tarsal conjunctiva and rubs it at least 4 times back and forth horizontally in order to collect enough epithelial cells. The end of the Dacron swab is then broken in a tube held by the porter, who records the references of the patient and the sample. The tube is kept cool, then frozen for transfer to the laboratory. The specimens taken during the field studies are conserved in the laboratory at -80 degrees centigrade until their analysis. PCR analysis requires complex instruments and procedures. It is performed in 4 separate and isolated areas. In the first, the reagents are prepared. In the second, sample DNA is extracted by robot in a protected zone. In the third area, the DNA is mixed with the reagent needed for amplification. In the fourth area, the amplification and identification of the target gene are performed with the help of an automatic thermocycler, PCR in real time, in a closed system dependent on marked probes that emit fluorescence. These quantify the chlamydial load of the sample. A sequencer allows for the determination of the sequence and thereby defines the strain genotype. Why bother with such a complex bacteriological test to diagnose a disease for which the clinical findings are so specific? Epidemiological studies of trachoma can be criticized by experts if they aren't substantiated by microbiological confirmation. The question being, is the clinical or the laboratory evidence sufficient, or should we have both? Regarding treatment, of course, what we want is to prevent blindness and interrupt the transmission cycle of chlamydia. But for a study starting in an unknown district or country, in which we see people with follicles, we should not jump to conclusions. The follicles could be due to an allergy or problem specific to a particular ethnic group or climate. At the start of the study, we took samples from subjects presenting clinical signs of follicular or follicular inflammatory trachoma. Over the years, the global cartography of trachoma established by the WHO has become more and more precise. The question was eventually raised about the trachoma situation in northern Cameroon. The first statistics concerning trachoma prevalence in Cameroon date back to 1955. A team of French physicians, led by Dr. Cousseran, organized the screening of inhabitants for ocular diseases in the region of the Logomé and Chari, north of the country. Out of 400,000 people examined, 1,600 cases of trachoma were found. In 1989, Dr. Yves Mage found a trachoma-related blindness rate of 4.6% in the ophthalmology clinics run by the NGO Ophtalmo Sans Frontières in the far north of Cameroon.
Il faut dire qu'il y a 20 ans,
it must be said that 20 years ago, OSF, a tiny NGO, had to deal with other public health problems, in particular cataract and a phenomenal number of these patients needing care. Nearly 1 person in 3 in our clinics. Then came the progressive training of our healthcare personnel that allowed our centers to become autonomous, and finally, now, being firmly established in the country, we have begun to address other public health problems, of which trachoma is one.
À d'autres problèmes de santé publique, dont le trachome.
In recent years, the healthcare personnel in the Kolofata OSF Centre have encountered a large number of cases of trachoma during consultations. The Kolofata Health District is situated in a very poor sub-Saharan zone where over 120,000 inhabitants are subsistence farmers and herders. The District Health Service organizes and coordinates all health-related activities, including public health campaigns in the field. The Kolofata Health District is one of the 28 districts in the Far North region. It consists of one district hospital, where we are now, which has 100 beds and offers all the services one expects from a reference district hospital. On top of the district hospital, there are 6 health centres, each one being the main sanitary unit for what we call a health zone. A health zone is an area well defined in terms of geography and demography, and has a health centre run by a nurse and his team of other nurses, community animators and village health workers who work alongside him in the villages. The smallest health zone has just over 7,000 inhabitants and the largest has 30,000. The district as a whole in 2010 is home to 122,057 people. All health centre and hospital offer curative care, that is to say, consultation of the sick plus surgery at the hospital, as well as preventive or public health care, vaccinations, prenatal consultation, educational outreach and occasional mass campaigns such as the trachoma treatment campaign. Needless to say, before any health intervention, one must prove statistically the existence of active trachoma in a region. This is why in 2006, Ophtalmos Sans Frontières requested a WHO expert to perform an initial prevalence study. There are 2 principal methods to determine if trachoma is a public health problem in a population. The first method is to perform a rapid evaluation. This is not an epidemiological study, but it allows one to rapidly assess if trachoma is a real health problem in this population. You go to the villages where you think the prevalence will be the highest. You take the first 50 children you encounter and examine them. You avert their eyelids and look to see if they have active trachoma. In this population, if you find more than 5 children with an active form of trachoma, you can assume that trachoma is a public health problem in the population. To have a much more precise idea of the real prevalence of trachoma in a population, you must do a study on a representative sample of that population. For this, you make a list of the health district's villages and randomly select certain numbers of clusters or villages and a certain number of children. In the case of the Kolofata Health District, there's a population of 120,000 inhabitants, and 35% of these are children 1 to 10 years old. From this population of children 1 to 10 years old, we randomly selected 2,400 children in 40 clusters of 60 each, and these children were examined for signs of active trachoma. This done, we only have to make our report. We have the prevalence of trachoma.
Un rapport et nous avons la prévalence du trachome.